Okay, folks, let's get started.
Apparently that's good.
How's everybody doing today?
Yeah.
I'm getting over a cold, so I feel kind of like that. Yeah, the best side of a cold is on the on the healing side, where you're feeling better. So I'm definitely feeling better about that, which is good. Can't think of any announcements or anything, so hopefully we'll just we're all set. We'll just dive into stuff. The web page issue did finally get fixed, and all the notes and all the videos and all that stuff are all now current and posted on the web page. So, as well as the grade distribution is also on the web page, and it's the same as the one that I emailed out to all of you. So hopefully you've all had a chance to look at that, and you know where you stand in the class. So, any questions about that, let me know. I want to finish up talking about blood clotting. Last time I got talking about blood clotting, and when I got at the very end of the lecture, I got to talking about prothrombin, and I tried to emphasize the important role that prothrombin had. Shh, I hear a lot of rustling. Okay. The important role that prothrombin had for basically localizing the clot to the site of the wound. So the way that this is done is that prothrombin, as I noted in class, is altered. And notice this is happening with the prothrombin, not the thrombin. So thrombin, of course, will be localized there as well, but the modification happens to prothrombin, not to thrombin. The modification that occurs is that, as I mentioned in class briefly at the end, glutamate residues at the end of, glutamate residues, that is the side chain of glutamate in the prothrombin, are modified. And they're modified by the addition of a second carboxyl group. So that second carboxyl group, if we look at it, looks like this. Okay? So here's glutamate, here's the side chain. The normal prothrombin only has one carboxyl group, but the modification adds a second one. Now this reaction, as I noted, uses an enzyme that requires vitamin K. So vitamin K is a vitamin that you can think of as a clotting vitamin. Okay? As a clotting vitamin. It facilitates the clotting process, and it does so by making this modification. So how does this modification then localize prothrombin to the site of the wound? Well, when you tear open tissue, one of the things that we'll talk about in a little bit is the fact that cells, calcium is really important for cells to try to control. Okay? Calcium is a very essential, it's a very important nutrient, but calcium in the wrong places in your cells will cause your DNA to precipitate. Not a good career move. So calcium is generally sequestered in cells, and as long as the cells are nice and tidy and organized, that's just fine. However, when damage or injury happens, those cells and that tissue are torn open, and that calcium is released, and it's not, in all cases, fully able to just freely go away, but it's actually attached to some stuff, as we'll see later. So the calcium is really an indication that something has happened at the site of this wound. These two guys right here, these two carboxyl groups, each of which has a negative charge, are very, very nicely positioned to bind calcium, which has a +2 positive charge. So they bind calcium that's localized at the site of that wound and hold prothrombin there. Prothrombin can then be converted into thrombin, and thrombin in turn can convert fibrinogen into fibrin, and the clot starts to form. Okay, so we see this sequential series of events. Well, what happens if you don't have vitamin K? If you don't have vitamin K, this modification does not occur. You only have one carboxyl group, one carboxyl group on the glutamate side chain of prothrombin proteins, and one carboxyl group doesn't bind calcium very well. Doesn't bind it very well. So as a consequence, prothrombin is not very well localized to the site of the wound, and bleeding occurs. Okay? Well, people have heard about blood thinners. All right? There's a variety of things out there that are described as blood thinners. Some affect this reaction and some don't. All right? So let me show you some that affect this reaction. If we look at blood thinners, okay, we see things like this. Okay, vitamin K is a pro-clotter because it's putting on that extra carboxyl group and allowing prothrombin to be localized to the site of the wound. Dicumarol and warfarin are both anti-clotters. They look like vitamin K. They are, in fact, competitive inhibitors of vitamin K's action, okay, because they resemble it in structure, at least closely enough as far as the enzyme is concerned. Now, people who've had heart attacks, people who have clotting issues, there's a fairly painful disease that occurs in legs called phlebitis, where clots form and cause problems. People who have these sorts of issues are given anti-clotting drugs, and they're given dicumarol, they're given warfarin, okay, in low doses. And it's important that they be in low doses, and it's important that they be titrated. That is, to determine the level that's necessary to prevent the problem. Because if you give too much of these, then they function in the same way that they function in rats. Warfarin is known as rat poison. Okay? And it's rat poison because it stops the clotting mechanism. The rat eats this, they bleed to death. And it doesn't mean they rupture open and they've got blood all over the floor. Most commonly, when you have very low levels of clotting, you're having internal bleeding and dying from that. Okay? So in a person, this level is monitored very carefully to make sure that you're not tipping over the edge to where you're going to favor, where you're not going to favor hemorrhaging, okay, because that would be a real problem, and get that level appropriate. Okay? Yes, Cassandra.
Is vitamin K actually used up in the reaction?
Is vitamin K used up in the reaction? The answer is it is not used up in the reaction, no. So it is a cofactor in the reaction used by the enzyme, but it's not used up. So it's not a substrate, no. Yes?
The change in the prothrombin that the enzyme with the vitamin K does that turn it into a thrombin, or is that a different?
Okay, so her question is, does the change that I just described of adding the extra carboxyl group convert it into a thrombin? And the answer is no. Remember that what's happening is we have zymogens, and with the zymogens we're breaking peptide bonds to activate them. So to activate prothrombin, we have to break specific peptide bonds. This reaction is putting carboxyl groups on, so there's no relationship between them. Okay? Good question. Yeah?
Does Draculin act the same way as
Does what?
Draculin?
Draculin?
It's a anticoagulant created by vector.
I don't know. There are other things people call, I've never heard of Draculin, so I don't know. Draculin sounds like a protein, I'm guessing, made by vampire bats. And, yeah. I mean, that's what it sounds like, right? I mean, when they put an IN, usually it refers to a protein. I'm guessing it probably does not work by that mechanism. We sometimes hear aspirin described as a blood thinner. People are on aspirin also to prevent excess clotting from happening. It doesn't work by this mechanism. So aspirin, for example, works by inhibiting the production of prostaglandins, and some prostaglandins are involved in making blood cells sticky, which helps in the clotting process. I would guess that's what that's working by, but I'm not familiar. I'd be curious. That's a cool name for a protein. I don't know. Send me a link. I'll take a look at that. That'd be kind of cool. Yes, Jessica. Is there a problem if there's too much uptake of vitamin K? What do you suppose? Yeah, I think if your vitamin K levels were high, you probably would have a problem. I believe your body will reasonably well regulate levels of vitamin K. It does that with vitamin D, for example. And it would be in the body's interest to do that. Because if you had too much vitamin K, what do you suppose is going to happen? You're going to start forming clots where you don't want. So that would be my guess, but I don't know that. Okay, so that's the story with the blood clotting and blood thinners. And I want to turn our attention last to talking about how we get rid of clots. All right. So clots are defense mechanisms to keep you from bleeding to death. But just like I said in class earlier, whenever we look at the body or we look at cells, we see that the body or the cells has a way of turning a switch on. They also have an equivalent way of turning the switch off. So just as the body has to be able to make clots, so too does the body have to be able to dissolve clots. And it does that. So that's what we're going to turn our attention now to, is looking at how we get rid of clots. Getting rid of clots is a function of an enzyme called plasmin, Plasmin. Plasmin will literally take that fibrin polymer that forms the clot. It will take that fibrin polymer and it will start digesting it, breaking it down into pieces so that the clot literally comes apart. In the healing process, removing that clot is an important step. If we had an internal clot in our body that formed, getting rid of that clot might be an important consideration. So being able to do that is essential for an organism. So the enzyme that does it is called plasmin. Plasmin is made as an inactive precursor. Surprise, surprise. It's floating around all in your bloodstream, and it's called plasminogen. So plasminogen is the inactive zymogen precursor of plasmin. And in order for plasminogen to become active, it must be activated. So guess what? There's a protease that cleaves plasminogen to convert it into plasmin. And that protease is known as tissue plasminogen activator, or as you see on the screen here, TPA. Okay? Now, TPA has a distinction, is that it was the first genetically engineered human protein to be approved for use in humans. Now, it wasn't engineered. It wasn't altered. The only engineering that was done was it was isolated from human cells, put into bacteria so the bacteria could make it, and we could grow a lot of this up. Tissue plasminogen activator turned out to be a real godsend for certain types of problems, one of which is shown here. Here is a heart with a blocked artery that you can see right here. The block occurred as a result of a clot. Treating this patient with tissue plasminogen activator broke down the clot and allowed blood flow, which is shown in white here, to resume. Now, tissue plasminogen activator is used a lot in patients who've had a stroke, for example. So strokes are commonly caused by clots in the arterial system that stop blood flow to some essential area. And it's been shown in recent years that if you treat a stroke patient who has had a stroke as a result of a clot, within the first 2 to 3 hours with TPA, you can reduce or eliminate many of the common side effects of stroke, including things like paralysis. So TPA is a godsend for treating people who have internal clots that have caused major problems for them. All right? So that's a very, very good benefit of genetic engineering. It's commonly used today. And again, it has to be administered fairly quickly after the clot has happened. I think, as I said, there's about a 2 to 3 hour time frame during which it's most effective. But during that time period, it's been shown that it does significantly reduce the level of problems associated with a stroke. Okay. Questions about that? Yes, Mark.
Are they adding, like, when they give patients TPA, are they giving increased levels of TPA than what we already have in our bodies to help break it apart?
Good question. Are they giving increased levels compared to what would be in the body? Well, A, the answer is yes, they are. But you can imagine there might be some problems associated with TPA. What do you suppose some of the problems might be?
Activates too much of that plasmin and starts waking up.
So you might see some of the effects like you see with warfarin. You might see hemorrhaging. You might see some internal bleeding, some things that would be a problem. So it's not given as a broadly used Band-Aid, but it's given for very specific purposes. And those purposes can, in fact, I mean, if you're weighing the side effects of a stroke compared to some internal bleeding, probably you're going to err on the side of the internal bleeding. That is, allow that to happen and reduce the effects of the stroke. Yes, sir?
Do they give it intravenously, or do they try to introduce it to the site of the clot?
Yeah, it's a good question also. The idea is that you want to target it as much as you can to the location of the clot. I don't know, and I'm not an expert in this, but my suspicion is that they probably would feed it up through the arterial system like they frequently do with the angioplasty or something. So they could target it specifically, because using it very broadly probably would have some other side effects, again, like hemorrhaging and so forth. But I don't know that, though. I'm not a physician. The effects of breaking down the clot can happen in the matter of hours. It's not an instantaneous thing, because as you probably know from having, like scab or something on you, that it doesn't happen overnight, but it can happen in a matter of hours to relieve that problem. And of course, once it starts breaking through just a little bit, you get a little bit of blood flow. That can make all the difference in the world. Other questions? Okay. That pretty much summarizes what I want to say about zymogens and protease activation of proteins. We turn our attention now to something that, as somebody pointed out to me before class this morning, this next stuff is fairly easy and it's fairly, I won't say fun, but it's things that almost all of you have seen before, because when you had organic chemistry, you had to memorize an awful lot of stuff about sugars. And guess what? You're going to do it again. So if you learned it last time, you're probably going to have it a lot better this time. Being able to talk this language is important. It's why they require you to memorize it here. It's why, in organic chemistry, it's why I require you to memorize it here. And so I expect that you're going to know these terms and so forth that I'm going to be throwing out at you. And as I said, they're not overly deep. Carbohydrates are, as we all know, compounds that we commonly refer to as sugars. There are complex carbohydrates, which are basically polymers of sugars, in some cases modified polymers of sugars. Or there are simple carbohydrates, which are, in fact, the sugars themselves. Okay? Carbohydrate derives its name from the fact that carbo refers to carbon and hydrate refers to water. And if we look at the structure of carbohydrates, I don't have it on there. If we look at the structure of carbohydrates, the formula for each of them can be written as CX, in parentheses, H2OX. Okay? For example, glucose is C6H12O6, which can be written as C with a 6 and then H2O with a 6. So it's a carbon hydrate. That's really what it is, and that's where carbohydrate gets its name from. Okay, so carbohydrates are also called saccharides. And saccharides derive their name from the fact that they mean literally sweet tasting. Okay? That's what a saccharide is. We'll talk about monosaccharides, disaccharides, polysaccharides. Monosaccharide being a single sugar, disaccharide being two sugars linked together, polysaccharides being many sugars linked together. Okay? There are two categories of sugars that we refer to. One category is referred to as the aldoses, and these are sugars that have an aldehyde structure in them when they're in their linear form. Okay? So an aldose, for example, is something that looks like these two guys on the right. Okay? Glyceraldehyde can exist in two stereoisomeric forms, the D form and the L form. And this is where you're going to thank me for being very simple with stereoisomeric descriptions, because we don't do the R and S, we do D and L. And you're going to see D and L is very, very simple. There's the aldehyde group. There's the aldehyde group. These two, this carbon in the middle in this case has 4 different groups that are attached to it. So it is, in fact, an asymmetric center and can therefore exist in 2 different forms. Okay? In contrast to the aldoses, and by the way, whenever you see the letters OSE on the end, it means a carbohydrate of some sort. So glucose is a carbohydrate. Fructose is a carbohydrate. Sucrose is a carbohydrate. These are all carbohydrates. On the other hand, instead of having an aldose, we can have a ketose. A ketose means that instead of having an aldehyde structure in it, it has a ketone structure in it. And what you're seeing on the screen are the simplest aldoses and ketoses. Right? To have a ketone, you have to have 3 carbons, because you have to have the carbon in the middle, surrounded by 2 other carbons, as you see here. You'll notice that in dihydroxyacetone, which is a ketose, that there is no asymmetric carbon. That's because the asymmetric carbon that we had in the other 2 was the middle one, but this guy only has 3 things attached to it, not 4. So this ketose only exists in one stereoisomeric form. All right. When we talk about sugars, we talk about them with respect to the number of carbons that they contain. So a triose is a sugar that contains 3 carbons. A tetrose is a sugar that contains 4 carbons. A pentose is a sugar that contains 5, hexose 6, heptose 7, octose 8, and we generally don't see sugars that have more than 8 carbons. We get that big, then we start seeing individual sugars linked to each other instead of seeing just one sugar of that size. Okay? We can combine these names. We could describe glucose, I'm going to show you in a second, as an aldohexose. Okay? An aldohexose, meaning it's an aldose and it's a hexose. You could call it a hexoaldose if you want to. I don't care. But the names, when you start putting these together, they all have to be right. It's an aldose and it's a hexose. So it's an aldohexose, it's a hexoaldose, whatever. Fructose turns out to be also a hexose, but it's a ketohexose because it has a ketone instead of an aldehyde. Okay. Now, let's take a look at some of these structures. Before I do that, I need to introduce one other term to you, and that is something called an enantiomer. If I look back at my structure of the D and L glyceraldehydes, these two guys are mirror images of each other. So two sugars that are mirror images of each other are known as enantiomers. Now I'm going to show you something about mirror images that's important in a minute, but I just want you to keep that definition in mind for the moment. That is that mirror images of sugars are called enantiomers. Okay. Now, here's the family, all right? And these are families. These are families of D sugars, all right? When we look at this, we say that if we have 3 carbons in it, and we start with a D, there's only one D sugar of glyceraldehyde. There's also an L form, and the L form would be over here with a different group. If we take that D form and we add another asymmetric center, we create a tetros, and we discover there are 2 tetroses, one called erythros and one called threose. Notice the structure of these guys. They're both called D sugars, and the common thing that they have is they both have an OH on the next to the bottom on the right position. The same thing down here. All of these guys are D sugars. They have the OH on the next to bottom position on the right. And the same thing down here. So a D sugar is something that has, in what's called its penultimate carbon, that is the next to the last carbon, it has the OH written on the right. Pretty straightforward. Now, if I were to say to you, okay, here is D-threose, what structure do you suppose L-threose would have? It would have the OH on the left, right? Would that be the only difference? Most people would say yes, including most graduate students and some faculty, but that's wrong. Turns out that the L sugar is the mirror image of the D sugar. So if I talk about L-threose, I've got to flip this to the left and I've got to flip this to the right. Okay? Don't forget that. So the L sugar is the mirror image of the D sugar. Not only does the last hydroxyl flip, but so does every other hydroxyl in it. Okay? Next, let's compare D-erythrose with D-threose. They're different stereoisomers. Okay? They're different stereoisomers. That is, they have the same formula, but they have different arrangements of things in space. They are not, however, mirror images of each other. So we have a name for them. We have names for a lot of things. Name for stereoisomers that are not mirror images of each other are called diastereomers. D-I-A-S-T-E-R-E-O-M-E-R-S. Diastereomers. So diastereomers are stereoisomers that are not mirror images of each other. Example, D-erythrose and D-threose. No, you're not going to have to memorize these structures. There's about 4, maybe 5 sugars I'm going to require you to memorize the structure of, and I require you to memorize them not to be mean, but because they are important sugars that you will use in other classes. I'll tell you what they are as we go through. Okay? Well, each time I add another asymmetric carbon, I add 2 additional possible structures for that, left and right, etc., as I go down. I discover at the bottom that I've got 8 different D aldohexoses. I also have 8 different L aldohexoses. So there's a total of 16. 16 is 2 to the 4th. 1, 2, 3, 4. The number of asymmetric carbons? 2 to the number of asymmetric carbons tells me the number of possible stereoisomers I have for a sugar. Okay. Now, on this figure, there are 3 structures I think you should know. Glucose, the most common sugar on the face of the earth. Mannose. Galactose. Galactose is probably the second most common sugar on the face of the earth. Okay? Now, they're related to each other. They're very similar to each other. And you'll see when we write them in the ring structure that they actually look even a little bit easier to figure out how to draw. Okay? So you should know how to draw these not only in the straight chain form that you see here, but also in the ring forms that I'll be showing you later. Okay, I'll emphasize again, these are all aldoses. Notice they all have an aldehyde structure at the top. The ketoses are going to be a little bit different, and we'll see that in a second. Questions about this before I go forwards? Anybody awake? Okay, let's look at the ketoses. We start with dihydroxyacetone, which did not have any stereoisomers. We add an asymmetric carbon, and there it is. This is going to be D-erythrulose, because the hydroxyl is on the right. Notice there's the ketone right there. I add another asymmetric carbon. Now I see 2 possibilities. I add another one. I see 2 more possibilities. And these are showing me, again, only the D sugars. The L sugars, I'm going to have 4 of those as well. So when I look at a ketohexose, I see 8 possible stereoisomers. And again, we have 1, 2, 3 asymmetric carbons. 2 to the 3rd power is 8 possible structures. There are 2 things, I'm sorry, there's 1 structure on here that I think you should know, and that is that of D-fructose. I forgot one in the last one. I'll go back and show you in a second. But D-fructose is, actually I said that galactose was the second most abundant. Probably fructose is the second most abundant, and galactose is the 3rd most abundant. Fructose is a component of sucrose. It takes glucose and fructose together to make sucrose. So fructose is very, very common sugar. Okay, now there was one other structure I forgot to mention before over here. It is actually a pentose, and that is ribose. You should definitely know the structure of ribose, but look how easy it is to draw. All the hydroxyls on the same side. It is a pentose. It only has 5 carbons. Ribose is important because ribose is the sugar that we find in nucleotides. ATP, GTP, CTP, UTP, TTP, not TTP, but UTP, all contain ribose. Okay? So, very, very important sugar. All right, now those are the basic monosaccharides whose structures that you need to know. Okay? Sugars, as I'm sure you learned in organic chemistry, are long enough. When they get long enough, the tail can start interacting with other parts of the molecule, and when they do that, they can form ring structures. In the case of aldoses, they can form hemiacetals, and hemiacetals occur when you combine an aldehyde, which is that terminal carbon, with an alcohol, which is one of the internalants, to make a hemiacetal. That hemiacetal is paired in the ketoses with as a hemiketal. A ketone plus an alcohol yields a hemiketal. Now, those are just names for the moment. If we look at the structures that are involved in these, back up. We look at the structures that are involved in these, we discover that the cyclization occurs as follows. Here's the linear form of glucose. And I think of glucose as right, left, right, right. That's the way I memorized it when I was taking biochemistry for the first time. Right, left, right, right in the open chain. This hydroxyl group right here is actually attracted to this carbon right here. It makes an attack on that carbon, and when it does that, it forms one of 2 possible ring structures. Why one of 2 possible? Well, what it's done is it has converted a carbon that was not an asymmetric carbon into a carbon that is an asymmetric carbon. Notice this carbon now has 4 different things attached to it. This one only had 3 things attached to it. As a consequence, we have to consider both of those possibilities. This newly created asymmetric carbon is called an anomeric carbon. Somebody asked me last time, I sang a song about anomeric. Now you know what anomeric is. An anomeric carbon is a newly created asymmetric center in a sugar. Newly created asymmetric carbon in a sugar. The anomeric carbon can exist in two different forms. The beta form, in which the hydroxyl group is pointing upwards, and the alpha form, in which the hydroxyl group is pointing downwards. Now, the top structure I could describe, if I wanted to, as an alpha-D glucopyranose. What is a pyranose? A pyranose is a sugar that occurs in a six-membered ring. Now, students frequently don't pay attention to this. I want you to pay attention to this. There are 6 members of this ring. And I think it's very useful and instructive whenever you write the structure of a sugar that you number the carbons, because then you can relate this to this. If you just simply memorize this and you memorize this, you don't have that relationship. Here's carbon number 1, 2, 3, 4, 5. And notice number 6 is sticking up above the ring. The 6th member of the ring is the oxygen. Okay? Make sense? All righty, assuming it does. If we do the same thing looking at fructose, what we discover is that in this case, the attack is again on the carbon with the double bonded oxygen, but the carbon with the double bonded oxygen is carbon number 2 in this case. When that happens, and they're only showing you one of the 2 possible forms, you see that this makes a ring structure that has 5 members. There's number 1, 2, 3, 4, 5 being the oxygen and wrapped around to there. Okay? There are the 5-membered ring. This 5-membered ring is called a furanose, F-U-R-A-N-O-S-E. I remember 5 as furanose. Let's number our carbons. Carbon number 1, above the ring. Carbon number 2 in the ring. Carbon number 3, 4, 5, 6 is above the ring also. 2 of the carbons are sticking above the ring. Notice that this is called alpha because the hydroxyl is down. It's the hydroxyl that determines whether it's alpha or beta, not the carbon. It's a very commonly mistaken error on exams. All right? The carbon doesn't determine the alpha or beta. The hydroxyl does. Can we have a beta fructofuranose? Absolutely. It's just not drawn on this figure for reasons mysterious and known only to the publisher. But yes, we see that. Cut through this like butter. Yes, question. Yeah.
What would the L be?
What would the L? So if we made the L, and I don't teach this, but I'll tell you here since you've asked. If you make the L, this last one, instead of going up, goes down. So that's the one that determines. Because again, remember it's number 5 here that determines that. So the orientation of 5 determines whether that goes up or down. Since we only deal with D sugars here, we'll only see that carbon going up. But good question. Okay, good. Ribose. Here's ribose. Ribose also exists in a 5-member ring. Carbon number 1, 2, 3, 4, 5. Only have 5 carbons in ribose. Deoxyribose is the sugar derivative that we find in DNA. Ribose is what we find in RNA. The difference between ribose and deoxyribose is a single oxygen. That oxygen right there is missing in deoxyribose. Okay? We never see free deoxyribose in cells. That doesn't matter for our purposes here. But next term, when we talk about how nucleotides are made, that will be a consideration. Okay, other questions? Yes, sir.
So the ribose, they're all the OHs are on the same side, straight chain.
Yep.
Why is it drawn pointing upward for the original structure?
This is the anomeric. The anomeric is the newly created one, remember?
What about the one
Over here? That's the D. So again, the D is pointing up.
always drawn up?
Right. So this is why I want you to count carbons. Okay? So when we count carbons, remember the 3 that were on the right were carbons 2, 3, and 4. All right? So here's carbons 2, 3, 4. Right? So right is going to mean that this goes up, and these two guys here is just simply pointing down. Make sense? Okay. So numbering the carbons, and his question is a good one. Numbering carbons will always bail you out in terms of I'm lost on this molecule. But if you go back to the structures and you look at the numbers, then they'll make sense. Yes, sir?
Instead of being confirmed that organisms that evolve in such a way that proteins become L form.
Have what?
What is L form?
That's for amino acids.
Unless so something
Yeah, see, there's nothing magical about L or D. Those are human designations. Okay, so there's nothing that means that an L or a D is there. Most sugars that we'll see are in the D configuration. Not all. We'll see some L sugars, and that's not there for adaptive mechanisms for the most part. It's just the way that they use sugars. But most of the sugars that we'll see are in the D configuration. If you look at some of the mirror images of some of the sugars that are sweet, the mirror images are bitter, which is kind of cool, I think. But there's nothing chemical about that. That's just the way that our receptors perceive them. Yes?
Why is the ribose not a furanose?
Why is ribose not a furanose? It is a furanose. I'm sorry?
It's like not.
They just didn't put it on there. But still, with any of these, you could call this beta D ribofuranose, just like the other one. So it is a furanose. So anything that makes a 5-member ring is a furanose. Okay? Now, if you've gone tediously through organic chemistry or even some biochemistry classes, they'll make you draw all the possible. For example, glucose can exist in a furanose form. Fructose can exist in a pyranose form. They're not common. So I figure, why bother with them? So we don't talk about them here. Ribose, you're only going to find that in a furanose form because there's no other thing for it to actually, that's not true. You can find it in a pyranose form, but it's almost never seen. Okay? So we're going to talk about the common ones here. That's important that we keep that picture. Yes, sir? Does DNA only use the beta form? In fact, you're correct. The only form of ribose and deoxyribose that we see is the beta form because the base on the nucleotides is attached right up here, and it's always above the ring. Always is. Okay. Let's talk about... there's a bunch of gobbledygook. This is simply showing you you're not responsible for these structures. This is just simply showing you what I told you a second ago. You can see, for example, fructose in a 6-membered ring structure. It's not very common. That's not important to us. What matters is these two forms. There's the alpha, there's the beta. Okay? So you can, for our purposes in this class, ignore the pyranose form of the fructose. You don't need to worry about these down below. But the other ones that I've shown you, the pyranose form of glucose, yes. Pyranose form of galactose, which you can figure out, yes. Pyranose form of mannose, yes. Furanose form of fructose, and the furanose form of ribose. Those you're responsible for. Okay. Steric hindrance is a consideration. Steric hindrance happens as a result not of DNL, but of relative position. Remember, these bonds can be twisted in various ways inside of molecules, and the twisting of those bonds can change angles. And in some of those angles, we can actually run into things bumping into each other. A real good example of this is something that happens in glucose called the boat and chair forms of glucose. Okay? These are the same isomer. And in fact, in this case, we're talking about the beta form of glucose. And the beta form of glucose can be written in two different ways. All right? Notice that this is looking like I flipped it into the alpha configuration, but I haven't. This is still beta. This is up and there's down. If I look at this form of glucose, the beta form of glucose, it's arranged in what we describe as a chair. Here's the back of the chair. Here's the legs of the chair. It's kind of a chase lounge sort of arrangement. The same beta form can have its bonds twisted so that it actually points more upwards, as you can see here. And when you do that, these guys start bumping into each other. This form is not favored. It doesn't mean it doesn't exist. It just isn't favored. 95% here, 5% here, something like that. Okay? This form is called the boat form because it looks like a little canoe. All right? So steric hindrance, as we talked about before, we try to scrunch things too close together. They're not going to fit very well, and they don't work very well. This is part of the reason we don't see the beta form of glucose as much as we see the alpha form. The alpha form is actually slightly preferred. Here's one you're not responsible for, but I'll show it to you anyway, and that is what happens in furanoses. Similar things can happen in furanoses. Personally, I find them a lot more difficult to think about in three-dimensional space. Boat and chair is a fairly easy designation to understand. These guys also have steric hindrance issues as they bend into certain ways, and that's what they're trying to depict for you here, but I find it's just not quite as simple to depict in your heads. So I won't make you responsible for that. Okay, oxidation. Another consideration in working with sugars is the fact that sugars have chemistry. Aldoses, an aldehyde group, is a fairly reactive group. So when we talk about aldoses like glucose, or ribose, or mannose, or galactose, those are all aldoses. What I hope you remember from your organic chemistry is that aldehydes are fairly reactive molecules. They're readily oxidized. When we oxidize something, it goes to an acid, as you see on the right. But I want to point out to you that that reaction only occurs when we have the molecule in the straight chain form. In the ring structure, we don't have an aldehyde. Well, when do we get the straight chain and when do we get the ring form? It turns out that in solution, glucose or any aldose is flipping. Straight chain to ring, back to straight chain. It's going back and forth. That means it can go from alpha to ring and back to beta, for example. It can go from alpha to ring and back to alpha. And the rule is, as long as this remains an OH right here, this flipping can happen back and forth and back and forth. If we put something on there, like let's say we take that hydrogen off and we put a methyl group on there, we create something called a glycoside. A glycoside is a sugar whose anomeric carbon has been altered in some way. So for example, if I alter the OH and make it an OCH3, I no longer have the ability to go back to the linear form. It's stuck in the ring. It can't flip back and forth because I said flipping back and forth required that to be an OH right there. Glycosides, once you've got them, will stay in that configuration. So if I put this guy right here and I put a methyl group on there, it would not be able to flip from beta back to alpha. Whereas if I didn't put that methyl group, it could flip beta, linear, back to alpha. So it can go back and forth. If I make a glycoside, it's stuck in that particular structure. Now, glycosides are not unusual things. Sucrose, for example, is a glycoside. Most polysaccharides are glycosides. We'll see how that occurs in a little bit. Now, the important thing is, however, with respect to oxidation, is that this guy can be oxidized. When it becomes oxidized, it goes to an acid. Wherever there's an oxidation, there's an equal and opposite reduction, because oxidation involves the loss of electrons. Those electrons have to go somewhere, and whatever they go to, they reduce. So in this case, the electrons are going onto a copper and reducing copper from +2 to +1. This involves a change of color. It's very easy for me to monitor the rate with which this change occurs by watching how the color of the solution changes that I've got it in. It's a very simple thing to do. Glucose and all aldoses are known as reducing sugars because they reduce something. They give up electrons readily. They, in turn, become oxidized. So they're oxidized. What they give their electrons to become reduced. So reducing sugar is something that is very readily oxidized. Ketones, you may remember from your organic chemistry, are not nearly as easily oxidized as aldehydes are. Ketoses like fructose, for example, don't get nearly as easily oxidized. Yes, it can be oxidized a little bit, but it doesn't happen very easily. It doesn't happen very often. So fructose is something that we describe as a non-reducing sugar. Okay? What are the rules? Okay. Can a ketose flip into a linear? You bet. The only difference is it's flipping with this second carbon, and the same rule holds. If we alter the hydroxyl on the second carbon of fructose, then it can't flip. It can't go linear. There's the glycosides. Yes, Megan.
The copper was just an example of oxidation, right?
The copper is just simply providing a means of oxidizing the sugar, so it's accepting those electrons. But you could have other things that could cause oxidation. That's correct. The reason we use copper is because of that color change, and it becomes very, very easy to monitor. So glycosides. Here's a glycoside, as I said. Methyl group on that oxygen. There's a methyl alpha D glucopyranoside. Wow, big mouthful of a name. Okay. There's the beta form. Once we get it in one of those forms, it's going to stay in that form. It's not going to flip. We also see what are called N-glycosides. In the case of N-glycosides, and we'll see some of these O-glycosides and also N-glycosides involved in blood groups, we see that the OH has been replaced completely by a nitrogen with something else attached to it. So if this is an NH2, for example, that would be an N-glycoside. It can't flip. It's not going to flip. The OH has been altered. Okay. Other examples. There's an L-sugar right there. Okay. L-sugar. This L-sugar fucose is not uncommon in our bodies, and you'll notice that again it's L, so the designation is it's going down instead of up on that penultimate. Some of these, like this guy here, we see commonly in what are called glycoproteins, and we actually see the protein. This is an amino acid or an amino acid-like compound. The protein is being attached through that bond right there, and so what we see are sugars being attached to proteins. Yes, sir.
Wouldn't that first one be alpha?
That first one would be alpha. You're right on. Yes. Nobody's ever pointed that out before. I've taught this for how many years? But yes, that should be an alpha. You're right. The book is wrong in places. Makes sense? Yes. So these guys, the question is, can they flip? Well, you look at the anomeric carbon. That's the question about whether they will flip or not. So if you have an OH on the anomeric carbon, yes, you do. That could flip. This guy could flip. This guy could flip. This guy could flip also. There's the hydroxyl right there. So yeah, all those could flip. Okay, let's see. How about a song? And that signals the end of the period. It's a real easy song. It just takes a minute, and it's actually based on what you've hopefully learned today. Ready? Carbohydrates all should sing, glory to the Hayworth ring. Anomeric carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L, diastereomer hell. Alpha, beta, D, and L, diastereomer hell. Thank you. See you guys later. You didn't join me here. You had your golden opportunity. How you doing, Andy?
Sorry, I came in early. I thought you were done.
No problem. No problem.
I don't like doing that.
I'd get you up here singing. I'll drag you up here next time. How you doing? Must be the middle of the term. You're getting ready to start your section, huh?
Yeah.
Let's see. Let's leave this on for you.
No, I'm going to
log it out. Okay. Okay. Hi. You have a question?
So vitamin K. So when a dog eats rat poison,
Yeah.
in the clinic, we give injection of vitamin K. And is that going to just cause that enzyme then to add the?
You're going to be competing with the rat poison. So you're trying to outcompete the rat poison. That's what you're trying to do. Make sense?
Yeah.
Because they're competitive inhibitors.
Got it. Thank you.
Yeah, sure.
We have a flipping question.
Flipping, okay. Let's go back here. Get out of the way here. How you doing? Good. Are you in here too?
Yeah.
Oh, you poor thing. Yes, what's up?
So when we talk about flipping, we talk about it flipping the OH and